The concept of epigenetic regulation refers to the various processes that can influence the activity of genes without altering the underlying DNA sequence. Unlike genetic changes, which involve modifications to the actual genetic code, epigenetic changes affect gene expression, that is, whether specific genes are turned on or off, through chemical tags that attach to DNA or histone proteins around which DNA is wrapped. This can have profound effects on cellular functions and is crucial during the development of an organism, as well as in the maintenance of cellular identity and function throughout life. Number 1. DNA methylation. This process involves the addition of methyl groups to the DNA molecule, typically at cytosine bases adjacent to guanine bases, referred to as CpG sites. Methylation generally suppresses gene expression by making it harder for transcriptional machinery to access the gene, thereby reducing its activity. In early development, DNA methylation patterns are reset. But as cells begin to differentiate into specific types, these patterns become tailored to ensure that only the necessary genes are active for a given cell type. 2. Histone modification. Histones are proteins around which DNA is tightly coiled. They can be chemically modified in various ways, including methylation, acetylation, phosphorylation, and ubiquitination. These modifications change the structure of the histone in such a way that either makes DNA more accessible to transcription factors, thus promoting gene expression, or less accessible, therefore repressing gene expression. During development, histone modifications help orchestrate the timely expression or suppression of genes necessary for proper growth and differentiation. 3. Non-coding RNA-mediated gene silencing. Non-coding RNAs, such as microRNAs, miRNAs, and long non-coding RNAs and cRNAs, regulate gene expression post-transcriptionally. They usually bind to the messenger RNA products of genes and can either block them from being translated into proteins or target them for degradation. This RNA interference mechanism is another way in which cells control gene activity without changing the genetic code itself. Overall, epigenetic regulation during development is essential for the intricate orchestration of gene expression necessary to form a complex organism from a single fertilized egg. It ensures that the right genes are expressed at the right time and location, corresponding with the developmental needs and environmental cues. Misregulation of epigenetic processes is linked to various developmental disorders and diseases, including cancer, pointing to the significance of epigenetics in health and disease. As scientists delve deeper into the realm of epigenetic regulation, it is becoming increasingly clear that the dynamic interplay between the environment and our epigenome can have significant implications for our development and well-being. Understanding how these sophisticated layers of gene regulation work opens the door to novel therapeutic strategies for many conditions that have eluded conventional approaches targeting the genome directly.